today. I haven't seen you since the day you went away. You made the world a better place.
Nothing in biology makes sense except in the light of evolution, and very little in evolution makes sense except in the light of genetics. We need genetical models to understand evolutionary change. In the mid-19th century, Gregor Mendel published his law of segregation, stating that alleles at a locus segregate independently during meiosis, and his second law of independent assortment. Alleles at separate loci are passed independently of one another from parents to offspring. His results were not fully accepted when rediscovered in the early 20th century. Mr. Udny Yule was one of the critics, arguing that Mendelian inheritance implied that dominant alleles would increase in frequencies in natural populations at the expense of recessive alleles. In the end, there would be only dominant alleles left, he thought, and since recessive alleles are found in natural populations, Mendel had to be wrong. Geoffrey Hardy was a British mathematician who published a science paper in 1908 showing that this criticism of Mendel was ungrounded. He thought this was pretty simple stuff, even for biologists. The question that Geoffrey Hardy asked was this: under what circumstances will the distribution of dominant and recessive alleles remain stable in a population? This question is quite easy to answer in bisexual species, where each individual carries both male and female reproductive organs. Consider a locus with two alleles, the dominant big A and the recessive little a allele. In a bisexual species, all individuals will produce male and female gametes at equal frequencies. If the frequency of the big A allele is P and the frequency of the little a allele is Q, we know that P plus Q equals 1. The probability of producing homozygous dominant offspring, that is, big A genotypes, equals the probability of randomly picking a big A allele from a male and randomly picking a big A allele from a female. Since allele frequencies are equal in male and female gametes, this probability equals P times P, or P squared. So P squared is the probability of homozygous big A offspring. Similarly, the probability of producing a homozygous recessive little a offspring equals the probability of randomly picking two gametes carrying the small a allele. This probability equals Q squared. There are two ways of getting heterozygous offspring, either by a male big A fusing with a female little a, or a male small a fusing with a female big A gamete. Both of these events occur with a probability P times Q, and the total probability equals 2 times P times Q. We say that for a one locus 2 allele model, P square, 2pq, and Q square are Hardy-Weinberg genotype proportions. So then, if the genotype frequencies in the next generation are expressed in Hardy-Weinberg proportions, what are the allele frequencies in the next generation after one round of random union of gametes? The frequency of the big A dominant allele can be found by summing the frequency of the big A homozygous genotype, that is, P squared, and half the frequency of the heterozygous genotype, PQ. The reason why we add only half the frequency of the heterozygotes is that only half the alleles carried by heterozygous individuals are of the dominant big A type. Another way of expressing this is as follows. The frequency of the big A allele in the next generation equals the frequency of the dominant homozygous genotype plus half the frequency of the heterozygous genotype. After some simple algebra, it is clear that the frequency of the big A allele is the same as it was in the previous generation. It is still equal to P. The same can be shown to be the case for the small a recessive allele. Also here, the allele frequency has remained constant. This shows that allele frequencies remain constant after one generation of random union of gametes in a population of a bisexual species. In the next lecture, you will see that this is also the case in unisexual species.